everyone, this is WAGATAM. So in today's video lecture, basically I'm not going to do any kind of board work because in today's lecture I'm going to talk about the putting sorting mechanisms. That is, all of us know that if I talk about a bacterium cell, then bacterium cell is having basically one compartment, and this compartment is surrounding by its cell membrane. But if I talk about a eukaryotic cells or a human cells, then our human cells or eukaryotic cells is having so many compartments. That is, like, we are having so many organelles like Golgi body, endoplasmic reticulums, mitochondria, nucleus, peroxisome, and so on. So it actually indicating that we are having lots of compartments, and all of us will also know that ribosome is called as an protein factory of our body, and inside a human cells there are near about 10 billion of protein presents, and a maximum of these proteins are synthesized from cytosolic free ribosomes. Now before this, I want to inform you that there are basically two types of ribosomes. The first type of ribosome is known as the cytosolic free ribosomes, and the another types of ribosomes is known as the membrane bound ribosomes. Now maximum proteins will form from this cytosolic free ribosomes, and the rest of the proteins will form from the membrane bound ribosomes. Now the proteins which form from the cytosolic free ribosomes, they are basically present into the cytosolic region of these cells. Now these proteins will have to pass from the cytosolic region of these cells to the different compartment or to the different organelles of our cells, so that the protein will have to perform their own functions. Got it? That means the protein will have to pass from cytosol to the different compartment. This is also called as a compartmentalization process. Got it? Now to pass this protein from one compartment to another compartment, this will have to follow total 3 mechanisms. And the first mechanism is known as the gated transport mechanisms, and the second mechanism is known as the transmembrane mechanisms, and the third mechanism is known as the vesicular mechanisms. Now in today's lecture, basically I'm going to talk about the gated transport mechanisms. Now if I talk about the gated transport mechanisms, then this is basically not an unidirectional transport. This is a bidirectional transport. That means the transport will before this the gated transport mechanism will occur between the cytosol and the nucleus. That means the protein which is right now present inside the cytosol, because it is just now synthesized from the cytosolic free ribosomes, and these proteins will have to pass into the nucleus, and from the nucleus there are also have to pass some proteins, that is like RNA and like mRNA and tRNA will have to pass from nucleus to the cytosolic regions. That means this is the bidirectional transport. The first things, that is the protein which is right now synthesized into the cytosolic region from the cytosolic free ribosome will have to pass into the nucleus, and from the nucleus the mRNA and tRNA will have to pass into the cytosolic region by using the same transport mechanism, that is known as the gated transport. So this due to this reason, this gated transport is known as the the bidirectional transport mechanism. Now I'm going to talk about the whole mechanisms. That is, let's suppose the protein is right now synthesized from the cytosolic free ribosome, and it is present into the cytosolic region of this cell. Now how it will pass into the nucleoplasm or inside the nucleus. Now whenever a protein will synthesize into the cytosolic region or any regions, at the times all of this protein is having a particular sequence of amino acid, and if any protein is having this sequence, that is proline, proline, lysine, 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 and proline, arginine, and valine. If any protein is having such type of sequence where lysine and proline will be present in maximum numbers, then such type of sequence will actually indicating that this protein will have to pass from cytosolic region to the nuclear regions. This sequence is actually known as the nuclear localizing sequence or the nuclear localization sequence. Got it? That means the protein which have this sequence will have to pass into the nucleus. Now those proteins who are having such type of sequence, this sequence is recognized by a special type of proteins that is known as the importin. Whenever importin will recognize this sequence, immediately importin will bind with this protein, and after that this importin will have to bind with special types of receptors that is present over the nuclear pore complex. Now what is the nuclear pore complex? Nuclear pore complex is a kind of the pore complex that is present over the membrane of the nucleus, and this is basically a octagonal symmetry and having 100 different types of proteins. So this is whatever the nuclear pore complex through which the protein and the protein along with importin will have to pass. Now this nuclear pore complex is having receptor for the importin. Now as soon as the importin will bind with its receptor that is present over the nuclear pore complex, immediately the importin along with the proteins that is synthesized into the cytoplasm of the cells will pass inside the nucleus or the nucleoplasms. As soon as the importin along with this protein pass inside the nucleus, immediately the dissociation will occur between importin and the protein. And after the dissociation, importin will have to bind with the GTP, or importin will have to convert into the active importin, so that importin can pass inside the nucleus back to the cytoplasm of this cell. Because importin is found the cytosolic region, but now this is present inside the nucleus, but it will have to be present into the cytosolic region of these cells. Now as this is present inside the nucleus, that means it will have to pass from inside the nucleus to the cytosolic region. Now to pass from inside the nucleus to the cytosolic region, the importin must have to convert into the active importin. Now how this importin will convert into the active importin? Inside the nucleus there is present the GEFs, that is the guanine nucleotide exchange factors. By the action of GEF, the importin will bind with the GTP, and as soon as the importin will convert into the active importin after binding with the GTP, this active importin will bind another transporter proteins that is known as the RAN. RNA is a special kind of transporter protein which will have to pass this importin from inside the nucleus to the cytosolic region of the cells. Now this importin and RAN complex will have to pass from the nuclear pore complex Got it? And as soon as the RAN and importin will pass into the cytosolic region of the cells, these active importins that was previously bound with the GTP will have to convert into the GDP, so that this active importin will convert into the inactive importins. Now how this active importin will convert into the inactive active importin? Inside the cytosolic region of our cells there is present the GAP. Due to the action of GAP, this active importin, which is previously bound with the GTP, now it's convert this GTP into GTP. Got it? And due to this region, the active importin will convert into the inactive importins. And this is how the proteins which is synthesized inside the nucleus from the cytosolic free ribosome will have to pass inside the nucleus, and after passing these ribosomes, the importins will have to pack inside the nucleus to the cytoplasm of the cells. I think this much is sufficient to know the gated transport mechanism